Hello and welcome back. We are continuing with Chapter 7, Microbial Genetics. In the last section, we had just finished up the different players that are involved in translation. And in this part 5 of the video, we're going to move on into actually seeing this process of translation. Roughly, it's separated into 3 stages. Initiation, that is getting things started. So getting things started means bringing together the messenger RNA, that small ribosomal subunit, and the large ribosomal subunit. Then elongation will start putting amino acids together. And then finally, termination is how it ends. We need a bunch of different proteins are making a play here. And then there's the energy that needs to be put in, and it's in the form of these triphosphate molecules. So initiation, you've got the small ribosomal subunit. There's that very famous start codon, the AUG. Every single species for every single protein, we all start with AUG as the start codon. In case it is helpful to you to think about it this way, back in the day when I taught freshman biology, I would always explain this to the students as, if you've ever started a roll of like tape, Scotch tape, there's that little tab, you don't need that for anything. Like the first thing you do is cut it off, but it's the place where you start. That's how I think about this AUG and this first methionine, this first amino acid that comes in there. It's like that start tab. So start here. And once you get all the players assembled, then we're ready. Like that's the initiation part. Get the 2 parts of the ribosome together, get that first transfer RNA with its methionine attached. Okay, and then we get to the next stage, which is, okay, start putting things together. So there's that first methionine. The next thing that has to happen is we need another amino acid, and which amino acid comes in is determined by what the next codon is. Once these 2 amino acids are next to each other, we pay using the energy from the triphosphate bond to put those 2 amino acids together to forge that polypeptide bond or that peptide bond. And then the whole thing can slide over. And then that empty transfer RNA can be released and gets recycled. But here we have now this growing polypeptide chain. That transfer RNA is attached to the P site. And then here comes the single amino acid, that's the A arrival, or A for amino acid, arrives there. Then we connect those together. And then again, everything kind of moves along. We've got this growing polypeptide chain. Here comes the next amino acid that's going to bind here. Ta-da! That's elongation. And how it's happening in a microbe, in a prokaryote, we've got that one messenger RNA. Now, here's a big difference, remember, between the prokaryotes and the eukaryotes. Because in the prokaryote, there is no nucleus, so this thing doesn't have to leave the nucleus to find the ribosomes. Even while we're making this messenger RNA, ribosomes can be connecting with it and starting the process of translation. I believe this is a typo, and that should say translation, not transcription. Okay, and then finally, termination. How do we know when to end this thing? Remember those 3 stop codons? UAA is one of those 3. Instead of binding to it just another regular old transfer RNA, here comes one of the so-called release factors. It's going to bind there, and basically, like, there's nothing to connect here, and so this causes all of these pieces to kind of pull away from each other and get released. Everybody goes their separate ways, and we have this polypeptide chain. Okay, and here's the little video that's going to go over these different parts: the initiation, bring it together, elongation, add amino acids, and then finally termination, let everything go, except now we have this strand of amino acids that's being represented here by these, looks like chains of pearls. So I'll play this, and then that'll be the end of this section. Oops, apparently I won't play this. Let me try that again.
There are 3 stages in the process of translation: initiation, elongation, and termination. Let's see how each stage works. The first stage of translation is called initiation. During initiation, the 2 ribosomal subunits, a tRNA with an anticodon complementary to the start codon, and several protein factors, assemble on the mRNA at the start codon. This results in an initiation complex ready for translation. The codon AUG is the start codon, which tells the ribosome where to start reading the message contained in the messenger RNA. In addition, AUG codes for methionine and formylmethionine at the start of bacterial transcription. The ribosome contains 3 binding sites for tRNA molecules: the A or amino acid site, the P or polypeptide site, and the E or exit site. The A site holds the new tRNA with an amino acid attached. The P site holds the previous tRNA with a growing polypeptide attached. And the E site allows the used tRNA molecules to exit from the ribosome.
This will make more sense when it's in action.
Transfer RNA molecules with attached amino acids are called charged tRNA molecules. Charged tRNA molecules carry amino acids to the ribosome. The anticodon on each tRNA is complementary to the corresponding codon on the mRNA,
Okay, so right
allowing
here.
for the translation from genetic code to amino acid. During elongation, the tRNA molecule with an anticodon corresponding to the next codon enters the A site. The ribosome catalyzes the transfer of the amino acid or polypeptide on the tRNA in the P site to the amino acid on the tRNA in the A site,
So connect these
forming
two.
a peptide bond.
Great.
After formation of this peptide bond, the ribosome moves along the mRNA to the next codon. This shifts the tRNA containing the amino acid chain to the P site, which was vacated by the initiator tRNA, which is moved into the E site.
So,
Now there's room in the A site for the next charged tRNA, and the process is repeated.
okay, so the bond forms and then the ribosome can move along. I think kind of like sewing machine. And then finally, here's the stop codon. Okay.
When the ribosome encounters a stop codon, a protein called a release factor binds in the A site, halting elongation. This process is called termination. The ribosome separates into its 2 subunits, and the mRNA and newly synthesized polypeptide are released. The ribosomes can then be recycled to continue the process.
All right. And then as just a quick note, this is a stop codon where it was supposed to occur, but remember that mutation that we saw, and you got a stop codon in a place where it should have just been another amino acid and continued translating. That's the big problem with those nonsense mutations. You suddenly have a stop codon like way over here, and that release factor comes in, and then everything gets released and stopped. So even though you have continued information in there, you're not going to get to translate it because that release factor came in and disassembled everybody. Okay, so that's the end of part 5. I'll see you over in part 6.